This is the pre-lab lecture for Experiment 8. Now, Experiment 8 is sort of the companion experiment to number 7, which you did this past week. In Experiment 7, you used a series of extractions and recrystallizations to separate the three major components of an Excedrin tablet. And as you saw, that while it looks great on the separation scheme, to separate the three components, it may have been a little rough in lab. So the second way that we oftentimes separate components in a mixture of materials is to do column chromatography. Now, if I take a step back here, the reason why we have to worry about separating the different components in a mixture is because it's easy to run an organic reaction. We boil things, we reflux them, which is a technique we haven't done yet, but in essence it's a boiling technique, or we mix things together. But the difficulty comes after we've done the reaction. We have to separate out the unwanted components and get the component that we want, and not only get it, but purify it and then analyze it. So column chromatography is a typical way that people will purify or separate components and purify the product of a reaction. So in this case, we're going to do the same experiment, or our goal is going to be the same as it was in Experiment 7, to separate acetylsalicylic acid, acetaminophen, and caffeine that are found in an Excedrin tablet. We're going to do this by column chromatography. The column chromatography experiment that we're going to do is similar to what you did with the spinach, except that we're going to collect a lot more fractions, and the spinach experiment was relatively straightforward because those pigments had a color and you could see them moving down the column. As you saw last week, the components of the aspirin or of the Excedrin tablet are colorless, and therefore we won't know when they come off the column. So we're going to separate these components, and in this experiment we're only going to use a half of a tablet to do this experiment. So here's an overview of what you're going to do. We're going to use a much larger column for this experiment. Not much larger, but it's going to be probably 2 centimeters in width instead of using a pipette. There are bigger columns that we can use, but in order to cut down on the amount of materials that we're using, we're going to go ahead and just use these columns, which I'll show you in a moment. So first thing we have to do is we have to prepare and pack the silica gel column. In the spinach experiment, what you did was you dry packed the column. In other words, you put the silica gel, or in that case we used alumina, into the pipette, and then you went ahead and you added solvent to sort of create the packing of the stationary phase, starting with dry stationary phase, and that's what most people call dry packing. The experiment that we're going to do this time is called wet packing. So we're going to create a slurry of silica gel in a chromatography solvent, and we're going to put that in the column, and we're going to go ahead and make a column of that silica gel. And it's important that that column does not have air bubbles, because if it has air bubbles, it'll create channels, and the solution will flow through the channels and not be separated by the silica gel. So that's an important thing to realize, is that our silica gel column cannot have air bubbles. It needs to be a very, very uniform layer. So once we prepare and pack the silica gel column, we're going to take half of an Excedrin tablet and dissolve it in mobile phase. And then we're going to place that solution on the column. Now, this column does not have a stopcock at the bottom, so that the solution will constantly be dripping out. It does have a glass frit, which is basically a porous piece of glass that's going to replace the cotton that we used in the spinach experiment. We're going to place a layer of sand on top of the frit to keep the silica gel from actually clogging it. We're going to have our silica gel, and then we're going to place a layer of sand on top to keep the silica gel from being disturbed. And then we're going to go ahead and place our Excedrin tablet solution on top, and then add mobile phase. And of course, when we add the mobile phase, Gravity is going to force the mobile phase down through the column, and as the components of the Excedrin tablet move through the column, hopefully they separate, and we can put compound A, the first compound to elute, off the column. This is A, and this is B, and this is C. Then the second column, or second component B, we can put in another test tube, and then we can put a third, the third component in the C. Now, we won't know what A, B, and C are for another week, and I'll get to that as I go through this. So we're going to add the mobile phase. Gravity's going to force the solution through the column. It's going to drip out, and we'll collect the fractions then in small disposable test tubes, just like you did for the spinach experiment. Now, the difference here is that the components of the Excedrin tablet are not colored, and so therefore we have to visualize them. And so to visualize them, we're going to have to spot a drop of each fraction onto a TLC plate, and then we're going to have to visualize the spots with a UV lamp, and I'll show you how that's done here in a few minutes. The idea here is to try and elute the three components into separate sets of test tubes. Then we'll combine test tubes together so that in the end we'll have component A in one sample vial, component B in a second sample vial, and component C in a third sample vial. We're then going to leave the caps off and allow the solvent to evaporate for a week so that we'll have our solids then, hopefully component A, component B, and component C in the three different sample vials. And then the following week, this is actually a 3-week experiment, extraction first week, column second week, and analysis third week. In the third week, we're going to determine the identities of components A, B, and C, and then we're going to look at the purity of the three components that we isolated, as well as the components that we isolated back in the extraction experiment as well. So these are the overall steps. We need to pack the column, then we need to basically separate, put the component, Excedrin tablet components on the column, separate those out, collect the fractions, and then analyze each fraction to see what, if anything, is in there. So here's what we're going to use. We're going to use 6 grams of silica gel, and it's important to actually weigh that out. You're going to make 100 milliliters of a 10-to-1 ethyl acetate hexane chromatography solvent. You're going to place that inside of an Erlenmeyer flask with saran wrap on top to keep the solvent from evaporating. We're going to use a slightly different solvent to develop the TLC plate. So you're going to prepare 10 milliliters of a 25-to-1 ethyl acetate acetic acid solution, and you're going to go ahead and place that inside of the chamber. Make sure that before you use the developing chamber, the developing jar, that it's cleaned with acetone and dried, and then when you put the solvent in here, you're going to go ahead and put the cap on it to keep the solvent from evaporating. I will provide you with some small plastic funnels that will actually fit into these small columns. It says to use a funnel that has a long stem on it. We're going to use a funnel like this, and this is not the same powder funnel that you have in your drawer, so there'll be a separate funnel for you to use. And if you wrote in your pre-lab assignment that you're going to use a long stem funnel, you can go ahead and make that procedural change that we're going to use a small plastic funnel. Then you're going to need to get a bunch of test tubes and the racks that are in the cabinets. We're going to use probably 20 or more disposable test tubes to collect all the fractions in this experiment. We'll use a chromatography column, which I'll talk about in a few minutes. It has a fritted glass piece here that replaces the cotton. And then we're going to use a little bit of sand to get a layer on top of the frit and then on top of the column. And this is not the sand that's just lying around in the labs. This is actually sand that's going to come from a bottle because it has to be relatively pure if we're going to do the column chromatography with it. So you're going to make these solutions. You are going to do this experiment in groups of 2 or 3, and every person is going to have a job. One person's job is going to be to actually collect the fractions. Another person's job is going to be to make sure that the solvent doesn't run out on the column. And if there's just two people, then the person collecting the fractions in the test tubes will also keep track of the column. And the third person will actually be spotting and developing the TLC plates. So it is a multi-person job with multi-person experiment with each person having an important job. So first step is to pack the column with silica gel. So you're going to weigh 6 grams of silica gel out into an aluminum dish and then place that inside of a 250 milliliter Erlenmeyer flask. Make sure that the Erlenmeyer flask is clean and dry before you put the silica gel into it. Then you're going to clamp the column, the small column here, vertically in your hood. And this column is probably 1 or 2 foot long. It has, again, this small glass frit at the bottom, so solution will come through the column. There's no way to stop that, so you have to make sure that something down here is available to catch the solution, and then you're going to place everything through the top. So first thing we need to do is to create the sand layer that occurs above the frit. So you're just going to take a little bit of sand in a scoopula and add until you get a 1 to 2 millimeter layer of sand above this glass frit. Okay, and the purpose of the sand is to keep the silica gel, which is much finer, from going into the glass frit and clogging it up. Then we're going to actually prepare the wet slurry that we're going to use to wet pack the column. So after you've prepared 100 milliliters of 10-to-1 ethyl acetate hexanes solution, and that's going to be basically 100 mil, well, the easiest way to do this is actually not to prepare 100 mils, but to prepare 110 milliliters, 100 mils of ethyl acetate to 10 milliliters of hexanes. You prepare that in your flask. Make sure there's saran wrap on it. You're going to pour in enough of that solvent to wet all of the silica gel that you just weighed out. And then you're going to go ahead and mix this to form a slurry with the stirring rod. And by a slurry, I mean it'll just be kind of a wet mixture of silica gel. Silica gel won't dissolve. And so we need to add enough to make sort of a slurry that we can pour, but not so liquidy that we're going to fill up the column. So once we make that slurry then, and you want to stir it to get all of the air bubbles out, you're going to go ahead and quickly stir and then pour the solution into the column using this funnel. And you want to make sure that when you pour the solution into the funnel that none of it spills out here, which it did in my demonstration, because all this solvent came because it spilled out of this powder funnel and then poured down the column. Now when you freshly make the solution and you pour it into the funnel, what you'll see is that the silica gel will actually begin to pack itself down at the bottom of the column. So as the silica gel settles out, we want to make sure there's no air bubbles, and so we gently tap the column with a stirring rod or a pencil in order to get this column of silica gel to pack evenly with no air bubbles. Okay. Here you can kind of see a better idea of that settling process. Here is the line that actually is between the settled silica gel and the silica gel that's settling from the solution. Eventually, this line will grow up the column until you get a nice separation of the liquid and the settled out silica gel, which now makes our column. And you can kind of see that better here. Here our column has now settled out, and the mobile phase is up here, and the mobile phase is now going to run down the column. And so we have to wait until the mobile phase gets close to the end, and then we're going to go ahead and add a little bit more of the sand to go ahead and create a sand protective layer so that as I put solution into the column, I don't disturb the silica gel. Okay, now you're going to probably not want more than about this much solvent in your column at any one time. So we added a few mils at a time, but you never let the column run dry. And by running dry, I mean that this layer of solvent actually drops below the top of the column, because at that point the silica gel will then get air bubbles and channels in it, and that's what we wanted to try and avoid in the first place. So once we have the column packed and ready to go, somebody's job will be to make sure they keep adding 10-to-1 chromatography solvent and keeping the column wet. Of course, what I'm not showing down here is the fact that we do have an Erlenmeyer flask collecting the solution as it drips out. Now, as the solution that you collect down here, you can reuse because it will be pure hexanes and ethyl acetate. Okay. So then what we need to do is then we need to prepare the Excedrin tablet solution to put on the column. So we're going to use half a tablet because the amount of silica gel that we have, we don't want to overwhelm it with components, because if we overwhelm it with components, then we won't get good separation. That was part of what we were trying to illustrate when we made our spots bigger and bigger in the spinach experiment, was the idea that at some point you can kind of have more component than stationary phase, and so you won't get good separation of the spots, and in this case separation of the components. So we take this Excedrin tablet and we're going to split it in half, and I have a bunch of pill splitters to do that. So we're going to split the tablet in half, and then one group can use one half and another group can use another. And then you're going to grind this up in these little plastic pill crushers that I have to create the powder, and then you're going to go ahead and pour that powder into a small beaker. Now, these are plastic pill crushers, and so I'm going to ask that you not clean these out during the experiment, because I don't want people to squirt acetone in here, because acetone will completely start to dissolve the plastic. So we don't use acetone. Just go ahead and leave these alone, and I'll clean them up at the end of each period. So we're going to just basically crush the tablet and then pour the tablet into a small beaker. Then you're going to add 1 to 2 mils of that 10-to-1 chromatography solvent in order to basically dissolve most of the components of the Excedrin tablet. Now remember, there's a binder in here that will not dissolve. So we're going to let this dissolve for a couple of minutes, and then you're going to stir this solution up and quickly pour it onto the column. And when you do that, the powder will settle to the bottom, and then we'll go ahead and allow the solution to drain down until it's just above the powder now, and then we'll continue to add mobile phase as the solution drains out of the column. Now at this point, you're collecting the solution into an Erlenmeyer flask, and it's going to take a few milliliters and a few minutes for this solvent to move the components down the column. So the solution that comes out now is going to be clean. It's not going to have any of the components in it. So when the solution drains just below the top, then that's where somebody will actually switch out the Erlenmeyer flask for a small test tube, and then the other person's job is again to make sure that this column does not run dry. Now if you position the tip of your column at the right height, you'll actually be able to slide the test tube underneath the tip, and it'll kind of hold it in place, and then the solution will drip into the test tube. And if you're saying that's not what's happened here, actually this solvent is still left over from the Erlenmeyer flask. So our solution will then drip into the test tube, and as it drips into the test tube, of course, the mobile phase is now moving down the column and hopefully separating our components. So what we're going to do is, we don't know when the compounds come off the column because they're colorless, and so it's not like the spinach experiment where we could see the yellow and the green pigments coming off the column to collect them. So as soon as the column, as soon as this solution reaches the top of the column here, and you've switched to your first test tube, We're now going to collect the test tubes until they're half full. So the solution will drip in. Once the solution fills the test tube half full, then you're going to go ahead and swap this for a brand new test tube. Now, in theory, the first several test tubes may not, they won't have any components in them. But we have to figure out the first test tube that actually has either acetaminophen, acetyl acid, or caffeine coming out of the column. So what we do is every half test tube, we switch it for a new one. We come over here and we put a spot of that solution onto the first sort of X of a TLC plate. Now, how do we know if there's any component in that test tube? Well, we then put that TLC plate under an ultraviolet light. How do you visualize spots that are colorless? Well, you have to use TLC plates that have a fluorescent dye that's actually attached to the silica gel. It's called a coating, and it's actually on the mylar, the plastic that is coating the silica gel. But what happens is that when we shine an ultraviolet light, and this is a 254 nanometer ultraviolet light. This is one that you might use if you're tanning. So don't be looking at this and just keep it pointed at the TLC plate, because this is a fairly rough UV UV wavelength. So as the UV light hits the TLC plate, what will happen is that fluorescent coating will glow. If there is a spot on the TLC plate, as you see here, it'll appear dark. If you think there's a small dark spot here, there very well may be, but I'm going to be looking for a fairly dark spot to indicate that that test tube actually had component in it. So what you do is you spot the TLC plate, and if you don't see any spot, any dark spot under the ultraviolet light, then that test tube doesn't have any component in it. So we continue this process. Every time a test tube fills up halfway, we go ahead and put a spot on the TLC plate. We put it under the ultraviolet light. If we see no dark spot, then we just continue on. The first time that we see a dark spot on a TLC plate, that's the first test tube that contains the component. And so we're going to call this spot number 1, and we're going to call that test tube number 1. Okay, so up to this point, no components have eluted off the column. But once we see that dark spot, then test tube number 1 has eluded from, or has compound 1 has eluded from the column. So from here on out, every half test tube gets spotted onto each lane, or it gets its own lane on a TLC plate. So you might have 3, 4, 5, 6, 10 TLC plates because you're going to spot every test tube from here on out. So this is test tube number 1. Test tube number 2 will be spotted here, 3, 4, and 5. And then on down the line. A new plate for every 5 or 6 spots, depending on how big we cut the plates and how closely you space your X's. Okay, so first time we see a dark spot, we now are collecting fractions. Okay, so after our first 5 fractions, we may see something like this, 5 dark spots. That indicates that all the test tubes then have individual components in them. And so now we need to develop the plate, which I'll talk about in a moment. But if you see a dark spot, that means that the test tube had a component of the Excedrin tablet. Now, if you think about what's going to happen, think about it this way. We have 3 components. As we run those 3 components through our column, in theory, we should have test tubes containing compound A, and at some point, there's going to be test tubes that don't contain any compound, because compound A has come off the column and now compound B is working its way down the column. So eventually you may see places where there's no components. So we've got our first component off the column, and now we're waiting, and now we see the second component coming off the column. So we may see these empty lanes indicating that those test tubes don't have any components in them. And in theory, you'd have compound A, then compound B, and then you'd see some empty lanes in compound C. Now, whether it works that way or not is dependent on how fast you push the solvent through and how much solvent we use, or how much stationary phase we use relative to the amount of component that we have. But that's what's supposed to happen. So once you get a plate spotted, then you go ahead and put that in your TLC jar that contains 10 milliliters of a 25:1 ethyl acetate hexane solution. And then that solution then will develop that plate. Now, the key thing is once you see the second component or the second set of spots start to come off the component, we'll assume that this is going to be component B. You're going to go ahead and switch your solvent then from 25:1 ethyl acetate to pure ethyl acetate when you start to see compound B come off the column. So when we develop our plates, this is theoretically what we're looking for. Since compound A elutes from the column first, it has to have the highest RF value on the TLC plate. That's how it made it through the column first. So you'll see some test tubes with compound A. You might see a couple of empty lanes indicating test tubes that don't have any component in it. Then when compound B comes off the column, you'll see then compound B spots, but they'll be distinguished from A because they won't have the same RF value. As a matter of fact, they'll have a lower RF value. That's why they come off the column second. Then you might see a couple of empty lanes, and then column C, the most polar of the components, then will come off the column, hopefully, and we'll see spots for that. So the initial spotting only tells you that there is a component or components in the test tube. It's the actual development that'll tell you whether it's compound A, compound B, or compound C. So when you start to see compound B come off the column, then you're going to go ahead and switch and start adding pure ethyl acetate, because that will help move spots B and C down the column faster. And then once all of compound B comes off the column, you're going to switch to pure methanol, which will hopefully elute component C off of the column. Okay, so this is how the developed plate should look in theory. It's possible that you may end up with a lane that has a spot of A and a spot of B. And so as you get your plates and your different developments, I'll be walking around, or Dr. Simmons will be walking around and looking at your plates to kind of see how your purity and how your separation is going. It's possible that you may have test tubes with just pure A. It's possible you may have test tubes with pure B, but you may in the middle have some test tubes that have both A and B in them. And if this was real research, what we'd end up doing is we'd end up taking those fractions and re-chromatographing them to separate A and B into their own individual test tubes at that point. Okay, so at the end of the experiment, you should have compounds A, B, and C in test tubes, and you should know which test tubes contain A, B, and C, or if you have mixtures, what test tubes have the mixtures by looking at your developed plates. So what you're then going to do is we're going to then combine the test tubes. And what I mean by combine the test tubes, pour the solutions from each of those test tubes into a tiered sample vial. And so you will actually put the test tubes that contain compound A into one tiered sample vial, and it's critical that it be taired so that way the next week, after all the solvents evaporated, you can determine the mass of the component that you've gotten. And then if you're going to put all of the test tubes that contain compound B into a second taired vial, and the ones that contain compound C into a third taired vial. So in theory, we've now separated components A, B, and C into the 3 different sample vials. Now we could rotary evaporate the sample vials to get the powders, but to be honest with you, there's really no rush to do that. So you're just going to place those vials after you've properly labeled them into a rack, and we're going to take the caps off, and we're going to let the solvent just simply evaporate over the next week, so that hopefully the following week we'll get just our solid materials, A, B, and C. Now, the problem with this at this point is we're labeling these as A, B, and C. I could conclude that A is the least polar of the components, B has medium polarity, and C is the most polar of the components, but I don't know what A, B, and C are yet. That's going to have to wait until the 3rd week. So at the end of the column experiment, you're going to place all of your waste solutions in the proper waste container because they'll contain ethyl acetate hexanes. The TLC jar will have contained ethyl acetate and acetic acid. You'll give the column to the instructor, or probably to me, and what I will do is I will then use a gentle air pressure to push the silica gel back out of the column, and then it can be cleaned and reused for the next lab period. You should sketch your TLC plates in your notebook, and what I mean by that is to create a sketch that looks like this, only now you have test tube 1 and 2 and 3 and 4 and 5 and 6, etc. on down the line, and you should determine the RF values for components A, B, and C, and you can do that simply by measuring them with a ruler and then writing that in your notebook. In theory, all of the test tubes that contain component A should have the same RF value. All the ones that contain B should have the same RF value, and so should C. Now, if it wasn't obvious from the spinach experiment, when you pull a TLC plate out, you need to make sure that you mark the solvent line. It's also kind of important to develop each TLC plate to about the same position, so that way when we line them up, we see the clear difference between A and B and C in terms of their RF values. Okay. So that's the end of experiment 8. Now, one experimental report is going to be turned in for both the extraction and the column chromatography experiments. So consider experiment 7, what you did this past week, to be week 1, the column experiment to be week 2, and in week 3 we're going to do the analysis of all of the different components that we got both individually and as a group to see how the purity of the components matched up in these 2 experiments. So between weeks 2 and 3, the solvents will evaporate from the vials, and so we should have solids of compounds A, B, and C in the sample vials. So in week 3, we're going to do the analysis of the components, not only of the vials of A, B, and C, but also your individual samples that you got from extraction that are in the aluminum weighing dish. So from the extraction experiment, you still should have the 3 solid active ingredients in the weighing dishes. If you didn't get a component, that's okay, you didn't get a component. And if the acetaminophen did not crystallize from the ethanol, then most of you have ethanol solutions of the acetaminophen that you can go ahead and at least spot on the TLC plates to determine what components are in there, and I'll talk about that in a moment. So you're going to determine the following data if you have the components to do this. You're going to determine the masses of all the isolated ingredients, if possible, and the melting points of only the acetylsalicylic acid and the acetaminophen. Caffeine doesn't melt, it sublimes. And so to measure the melting point of caffeine, you actually have to seal the capillary tube, and so we're not going to go to that trouble. We're just going to measure the melting points of the acetylsalicylic acid and the acetaminophen, both from the extraction and chromatography or column chromatography experiments, if you have enough to actually determine the melting points. Then we're going to determine the purity by doing TLC. So we said back in the spinach experiment that you could use TLC to determine the purity of a component to see how many spots do you get. Well, we're going to look at the purity of each of the components or each of the compounds that we isolated from our extraction and our column experiment. And we're also going to determine the identities of compounds A, B, and C from the column experiment. And we're going to do that by comparing to authentic aspirin, Tylenol, and caffeine tablets. So I will have an aspirin tablet crushed up, acetaminophen, which is in Tylenol, and a caffeine tablet all crushed up and dissolved in solvent in little vials so that you can go ahead and place authentic spots onto your TLC plates. So we're going to have two plates here, one for the extracted experiment and one from the column experiment. So you're going to place a spot of authentic acetylsalicylic acid in column maybe A, authentic acetaminophen and authentic caffeine spots in the first three. Then you're going to, from your aluminum dishes, you're going to take a little bit of solid, that's on the next slide. You're going to prepare the samples for TLC analysis by taking a small bit of solid, putting it into a disposable test tube, and adding 5 to 10 drops of the chromatography solvent to dissolve that. Then you're going to place a spot of that solution on the TLC plate. So you're going to do this for the extracted acetaminophen, the extracted acetylsalicylic acid, extracted acetaminophen, and extracted caffeine. You're going to do the same thing, preparing authentic spots for the column experiment, and then spots of, in this case, you don't know which is which, so you're going to have column component A, component B, and component C. So you're going to place those spots on there, and by comparison of the RF values, you should be able to tell whether A was acetylsalicylic acid, acetaminophen, or caffeine. So when you do this experiment, then you're going to see, and you may see the spot of acetaminophen come this far, maybe or acetylsalicylic acid, maybe acetaminophen goes here, maybe caffeine goes here. I don't know what the values are, or I've forgotten them, so I'm just using, these are not necessarily correct values. But then what we would see is, in the case of the acetylsalicylic acid, if we have it, it'll have a spot at the same RF value. If we find any other stray spots in here, it'll tell us that our sample's impure. And then for acetaminophen, we should see an acetaminophen spot. And then do we see any of the other components in there? For the caffeine, the same thing. We should see a spot corresponding to the RF value of caffeine. And so that's how we're going to determine the purity. If you see more than one spot, then that tells you that the sample wasn't pure. It'll also help you make the identities, because it could be maybe some caffeine got into the acetylsalicylic acid, so you might see a caffeine spot in there as well. So you're going to use the number of spots that you see to assess the purity of the extracted components. In the case of the column experiment, where you've placed spots of column purified A, B, and C, or separated A, B, and C, the main purpose here is that we determine which component is A, B, and C, because you'll have an idea about the purity from the previous TLCs that you ran the week before. So again, you're going to place a spot of acetylsalicylic acid, maybe acetaminophen, maybe caffeine. Again, I don't know what order they come out in, so we'll find that out. But then the question is, A should then match up with one of those three. Maybe B will match up with this one, and maybe C will match up with this one. So the idea is from the authentic RF values, that's going to help us determine then what the components A, B, and C identities are. So that's what you're going to do in week 3. Measure melting points, measure masses, do the TLC plates, and then develop the TLC plates. And you'll do these TLC plates individually, even for the group experiment. So you're going to use the TLC plates to assess the purity, and you should sketch out those TLC plates in your report and in your notebook, or I suppose you can take pictures of them. I guess there's one thing that I forgot to mention along the way, and that is that when you actually develop a TLC plate, you're going to see one of these dark spots maybe occur up here. Now, unless you have one of these tanning lights at home that you can stick these TLC plates under, and if you do, don't tell me that you do, but you have to take a pencil and circle the spot so that way later on you know where the spot ended up. Okay, that's one thing. We didn't really have to do that in the case of the spinach because they had a color to them, but in this case we have to circle the spots with a pencil in order to know where the spots were when we don't have access to this light. So you're going to include sketches of your developed plates in your notebook, and then I'll ask for them in the report, and then you're going to create tables of RF values and RF values and identities for both the for both the column experiment results as well as the extraction experiment results. And then also in the report and in your notebooks, you should include a sketch of the TLC plates that you obtained during the column experiment. So where I've shown you here sort of what I would expect to be the results, you want to make a sketch of this in your notebook, and then you'll make another sketch of it in the lab report that you turn in. And don't copy and paste mine when you put it in the report. Sketch what yours actually look like. Okay? Now there's one thing that happens along the way that I'll save for last, and that is that depending on how well you pack your column, it may take a considerable amount of time for your solution to flow through the column. Some people's columns drip very quickly. Some people's columns drip not very much at all. So in order to force the solution, in order to give it some pressure and move it a little bit faster, we have designed this little device that will allow you to push the solution, in essence, through the column. Now remember, you get the best separation with a slow column movement or slow mobile phase movement, so the faster you push, the worse your results will be, but if you wait forever, you will get no results. So we have to find a happy medium in between. So here's the device. You're going to take the stopper and put it snugly in the end of the column. Then that tubing goes to what's called a three-way glass Y. Y because it looks like a Y. And then we're going to attach one of these rubber bulbs to one end, and the other end is open. So to push the solvent through the column, number one, we fit the stopper firmly on the end of the column. Yeah, we're going to do that, and then occasionally we have to take it off and put some more solvent in and then put it back on again. Step two, you're going to take your fingers and pinch off that tubing so that when, in step three, I take my hand and squeeze this bulb, that the pressure will actually push the solvent down the column. Okay. So you want to squeeze the bulb slowly to push the solvent down. And this is why this is a more than one person operation, because when you have to do this, it takes one person to carefully push the column and then another person to collect the samples and basically say to stop because your test tube is half full. So how do you stop? Well, the second part of this is the pushing part. After you squeeze the bulb, you have to be careful not to suck the solution and suck the silica gel back up into the back up the column, because then we'll get air bubbles that we tried so hard not to get from the beginning. So after you're done pushing some solvent through, you then release this tubing so that then when your hand releases this bulb, it doesn't create a vacuum that sucks the solution up. It just merely sucks air in from this piece of tubing. So you have to use this process in order to push the solvent. So pinch, squeeze, and then release the pinch and carefully release the squeeze of the red bulb. And if you do this, you'll be able to push the solvent through at a little bit faster rate than sometimes the solvent will flow with gravity. Okay, and so you will probably have to do that at some point during the experiment in order to be able to finish. So it's important that somebody do this and that they don't suck the solution back up into and break our column that we worked so hard to make. Okay, so that's our pre-lab lecture for experiment 8, the column chromatography experiment. And so, of course, this is a group experiment, and we'll be going through this along the way. But hopefully this gives you a sense of what you're going to do and what you're going to see for this experiment.